Here we are moving on to the muscular system, Chapter 6, Lecture number 2. Muscle cells have some special functional properties that enable them to perform their duties. The first of these is irritability, also termed responsiveness, which is the ability to receive and respond to a stimuli. The second, contractility, is the ability to shorten when adequately stimulated. This property sets muscle apart from all other tissue types. Extensibility is the ability of muscle cells to be stretched, whereas elasticity is their ability to recoil and resume their resting length after being stretched. To contract, skeletal muscle cells must be stimulated by nerve impulses. One motor neuron, which is a nerve cell, may stimulate a few muscle cells or hundreds of them, depending on the particular muscle and the work it does. One neuron in all the skeletal muscle cells it stimulates is called a motor unit. Portions of two motor units are shown. The motor neurons reside in the spinal cord, and their axons extend to the muscle. Within the muscle, each axon divides into a number of axon terminals distributing to muscle fibers scattered throughout the muscle. This figure shows a photo of an actual motor unit. Now, when a long thread-like extension of the neuron, called the nerve fiber or axon, reaches the muscle, it branches into a number of axon terminals, each of which forms junctions with the sarcolemma of a different muscle cell. These junctions are called neuromuscular junctions. You can see here an axon terminal of a motor neuron forming a neuromuscular junction with a muscle fiber. Although the nerve endings in the muscle cell's membranes are very close, they never touch. The gap between them is called the synaptic cleft, is filled with tissue, fluid. It's called interstitial fluid. The axon terminal contains vesicles filled with the neurotransmitter acetylcholine, which is released when the nerve impulse reaches the axon terminal. The sarcolemma is highly folded adjacent to the synaptic cleft, and acetylcholine receptors are present in these folds. The axon terminal contains vesicles within the neurotransmitter acetylcholine, which is released when the nerve impulse reaches the axon terminal, as I have said. The sarcolemma is highly folded adjacent to the synaptic cleft, and acetylcholine receptors are present in these folds. Then the sarcolemma becomes permeable to sodium, which rushes into the muscle cell, and to potassium ions, which diffuse out of the cell. However, more sodium enters than leaves. This imbalance gives the cell interior an excess of positive ions, which reverses the electrical condition of the sarcolemma. This event, called depolarization, opens more channels that allow sodium entry only. This upset generates an electrical current called an action potential. So here you can see the acetylcholine diffusing across the synaptic cleft and attaching to receptors on the sarcolemma. This initiates the changes in the electrical condition of the sarcolemma. As we stated, the sodium rushes into the cell, generating an action potential. And once begun, the action potential is unstoppable. Got to remember that. It travels over the entire surface of the sarcolemma, conducting the electrical impulse from one end of the cell to the other. The result is contraction of the muscle cell. Here we can see a comparison of the action potential to a flame consuming a dry twig. Right? So you light the flame, and it's going to proceed all the way to the end until it burns itself out. Now, what causes the filaments to slide? This question brings us back to the myosin heads that protrude all around the ends of the thick filaments. When muscle fibers are activated by the nervous system, as just described, the muscle heads attach to binding sites on the thin filaments, and the sliding begins. Each cross bridge attaches and detaches several times during a contraction, generating tension that helps to pull the thin filaments towards the center of the sarcomere. As this event occurs simultaneously in the sarcomeres throughout the muscle cell, the cell shortens, which causes the contraction. Now, the top figure shows the muscle at rest, while the bottom shows when the muscle is fully contracted. Notice that in the contracted sarcomere, the light H zone in the center of the A band disappeared. The Z discs are closer to the thick filaments, and the I bands have nearly disappeared. The A bands move closer together but do not change in length. Got to remember that. In skeletal muscles, the all or none law of muscle physiology applies to the muscle cell, not to the whole muscle. It states that a muscle cell will contract to its fullest extent when it is stimulated adequately. It never partially contracts. However, the whole muscle reacts to stimuli with graded responses, or actually different degrees of shortening. So in general, graded muscle contractions can be produced two ways. One, by changing the frequency of muscle stimulation, and two, by changing the number of muscle cells being stimulated at one time. We briefly describe a muscle's response to each of these in our next lecture. So let's take a look at the key points here that we have for this particular lecture. Number one, what two structures are closely associated at a neuromuscular junction? What ions enter the muscle cell during action potential generation? What chemical, ATP or calcium, triggers the sliding of the muscle filaments? And our last question, which is a cross-bridge attachment more similar to? A precise rowing team or a person pulling a bucket on a rope out of a well? All right, this ends this lecture. We're going to move on to the next one.